I'm going to be talking about new talk. Actually, I call it good kmers and bad kmers in viral metagenomics, and that'll make more sense in a little bit. What I'm actually going to do is start my talk by kind of reviewing where I left off last year and to kind of highlight what we focus on this year. So the goal is we really, in the lab right now, is that we want to develop a inexpensive, scalable diagnostic test that can identify any respiratory pathogen. We call this the universal metagenomic diagnostic. And we're thinking like something that could do like 5,000 tests a day at $20 per test. So it's so inexpensive that instead of getting a COVID test, we would have a test that would just give us everything, information on everything, and it would be inexpensive enough where like the same cost that we spend now on a COVID test, but we think that would really change how people think about infectious disease, and it's also, as I'll emphasize in the talk, I think it's really within reach of where we are now, and there's a lot of challenges to this: logistics, IT, RNA-seq part, bioinformatics databases, and FDA and regulatory. And so we've made progress, I think, on a lot of these. And in this year, we actually made a lot of progress on the bioinformatics part, which is what I'll be talking about. And just overall, kind of how this fits in the lab. Like we, in 2020, we developed a COVID test, which is a sequencing-based targeted diagnostic. And so that really worked well. We ended up doing close to 2 million tests. And then we switched to try to develop a what we call 2.0, which is like this metagenomic test, which is what I'm really going to be talking about today. This is really it's called an NGS agnostic diagnostic in the kind of diagnostic community, but we would call it a different name. We would call it RNA-seq. So this is basically an RNA-seq based technology, right? And it's actually working well. It's expensive. It's actually probably closer to $100 a test. And that's what I'm talking about. And then our plan is to to make a much less expensive version of it. That would be just on the technology side. And so, but then in parallel, we're also working on a targeted test that could detect kind of any, like every known pathogen that's circulating for much lower cost as well. So, but today I'm really going to be focusing on this kind of RNA-seq based. And so, kind of what's new? This is actually a slide from a couple of years ago, but I think the situation, I think this really needs to be emphasized because people previously have really been talking about how sequencing is expensive and the cost of the sequencers, is really the prohibitive aspect to the test, to any kind of diagnostic or even any kind of sequencing analysis. But that's completely changed. I think that this new generation of Illumina sequencers is really transformative. The cost per read is so low now that sequencing is no longer the bottleneck in cost. I mean, in fact, the cost is really staff and reagents for the library preparation, right? So this is a new thing, right? So like in our test, for example, the library preparation is probably close to like the one version is like $70 and then the sequencing is like $10, right? So that's a crazy ratio. So all the challenges now are really focused on, figuring out how to make the non-sequencing parts more expensive. And so this really requires rethinking approaches, and things like getting more depth and things like that is not an issue. Okay, so just to take a step back, when we talk about pathogen detection, there's really kind of two major paradigms. So the most commonly approached, if you go to get a test now, it's basically a PCR test. And what does PCR do? So PCR basically is a reaction that will amplify a specific target of RNA in this case, and so it's got these primers that have sequence that has an affinity to the specific sequence. And so if that sequence is present in the sample, it'll get amplified to the point that it pretty much dominates the sample. And so then either using kind of fluorescent imaging or sequencing, the whole sample is that RNA, and that's how you can detect that the virus is present. This has been the standard of care for more than 2 decades, and it actually makes a lot of sense because you don't need that much of an ability to detect the virus because your whole sample is essentially that RNA. So it's relatively inexpensive. You can even these like fluorescent imaging techniques can actually see it essentially. So that's what PCR. The only problem with PCR is that you have to know ahead of time what you're looking for. So that has 2 issues. One is that things that are completely unknown or very rare, you're not going to be able to see. But even more practically, it's very hard to do PCR reactions with many, many different primers. And so they interact with each other and they become less efficient. So it's kind of, you have a limit to how many things you can detect, even if you want to detect all of them. So in a different line of work, we're working on some strategies for that. But so then another approach is what's called depletion, which is basically if you try to, almost everything is human RNA in the sample. If you try to knock out the human RNA using, like there's enzymatic approaches or like CRISPR even, if you use CRISPR targets for human, you can try to knock out all the human RNA, and then what you're left is the viral RNA, and you sequence that, and so that's another approach. And then there's another approach, which is just sequence a lot. So just sequence everything. That's actually what we do. We started looking at depletion, but then because of, like what I mentioned, that the sequencing is no longer a bottleneck. And we also found that the sequencing, the quality of this viruses in general have very low quality. And so the quality is affected by reaction and library construction. And then if you add that you're also doing depletion, it oftentimes damages what little virus you have there. And so then you actually lose signal, even though you're more efficient to find it if it was there. So that's what we found. But so anyways, the basic paradigm is just that, we take the sample, we just sequence it, RNA-seq, and we're going to look at try to find these very rare signatures of viruses in the RNA-seq, and that's how we're going to have our diagnostic, even though we know that 99.9% of all the sequence will be human or junk or whatever, but that's fine. We'll just sequence enough to see what we're looking for. Again, the challenge was that this is just some plots from the lab showing that there's just extremely poor quality of the RNA. So oftentimes, when you do a sequencing experiment, you create your libraries and then you actually quantify them. We actually very often the samples are unquantifiable using these instruments. So you actually can't see that there's any RNA there, but then when you sequence, you actually see something. So, the limit, these are very poor quality samples. But on the other hand, some people in our community here have worked on, sequencing Neanderthals and other ancient DNA. And so, this has to be an easier problem than ancient DNA, right? So we're always saying, if they can find Neanderthals, we can find a virus in a sample, right, that PCR could find, right? So that's kind of been our mantra, knowing that there's something there, just a matter of time. And so this is what I mean by low quality RNA. So I don't know if anyone's ever seen a sequence trace like this, if this tells you what's actually going on. So essentially, this is a plot showing the frequency of amino acid, of nucleic acid per position. And so you can see that it's almost 100%. So basically, this is kind of a very funny looking data in the sense that all the reads almost all look exactly the same, right? For a whole flow cell. That's just because you know, we were getting almost no RNA, and so it was just the adapters kind of sequencing, right? So this is the kind of thing we did, and so essentially we did about 1,200 samples of optimizing the sequence, and it went from very bad data to just bad data, and so this was a success, but it was good enough for us to get going. And so this is actually what our results, our kind of diagnostic looks like. So these are actually samples that were, each row is a sample, and these are samples that were submitted for COVID testing. So we have kind of a PCR result for whether they're COVID positive or negative, and this is just kind of, these counts are the number of reads that fall into these bins. And so in this first sample, for example, we see that there's about 1.425 million reads. About 1.35 million of them are classified. Classified means that we can kind of classify them into some kind of a species. 1.3 million are human, 41,000 are bacteria, 2,400 are virus, and 2,100 of those are SARS-CoV-2. And so then we would say that this is we actually would call this a positive, and in fact the PCR result was positive, so they're concordant. So I'm just giving you this example just so you understand the scale, right? So you're looking through 1.4 million in order to find these 2,100, and this is a relatively strong signal. Like here's actually a positive with only 104, but the interesting thing is that, for example, this individual was, the COVID tested negative, but they clearly have RSV, for example, and this is like one that has a very, it's negative, but maybe they had a little bit of COVID. So anyway, but that's just a result. I just want to give a sense of scale. So what we did, we actually showed, this is 2 years ago, we showed that our test is very concordant with the PCR for COVID, which is, because we had that data handy. But the interesting thing is that we also started to find many other viruses in our samples. But of course, at the time, we didn't, we only had positive and negative results for COVID, so we didn't know about any of these. But we saw like a lot, maybe when this is basically additional 0.5x have an infection with a different pathogen. What I'm saying is that let's say our positivity, because it varies, but this kind of stayed pretty constant. So let's say our positivity was 4%, on that day. Another about 2% would have something else that we would see. So this is very promising. And so what we then did was we collaborated with the health system, and we got 100 clinical samples for them, which were, basically, they collect lots of samples from patients that have respiratory disease, and not every sample that they run, but they run about 7,000 to 10,000 samples a year in what's called a respiratory panel, which is a device. The one that they're using is called an E-Plex, but it's basically a device that does PCR on about 20 targets. It's kind of like a cartridge-based system, and it's very expensive actually. And so they ended up giving us 50 positives and 50 negatives, and these are the viruses that they find, which is like this is adenovirus. These are the different coronaviruses, non-COVID, human metapneumovirus. This is enteroviruses, parainfluenza virus 1 through 4. So these are the ones. There's many different vendors that sell a device like this, and they all pretty much find the same viruses. So that was successful. We analyzed another 400 and basically analyzed about total of 460 samples, and this is what we found. We found basically, for a lot of these, with the exception of enteroviruses, the results are really good, and very concordant with their devices. And there's there's reasons why we don't expect to see 100%, and we'll kind of talk a little about this later. But the interesting thing, at least at the time, was that we also found other viruses in the samples, especially in the negatives. So I think we saw a couple of these. So maybe, close to 1% or 0.5% of these patients had influenza C. And so maybe of the negatives, probably like close to 1%. So, in Wikipedia, I looked it up because I didn't even know at the time that there was an influenza C. I've never heard of it. It says it's not infectious in humans. So I was like, So, I mean, it might actually be generally not infectious in humans, and people are oftentimes immunocompromised and then getting these tests, and so it might actually be somebody's immunocompromised, which is why they have influenza C. But we see it. But the real question we were wondering was did we really see influenza C, or is this some kind of an artifact? And so the way that we addressed this was we also have another bigger sequencer, and so we basically just sequenced really deeply our same samples, and then we assembled these reads, and so the idea is that if we do this assembly, some of these contigs will correspond to the viruses, and that'll give us kind of an overwhelming amount of information to see what's going on. So here's what the results were. So the results were, we got all these assembled contigs. We got 173,000, about 100 million in total length. Why is that only? That's not a lot, but that's because we're only looking at RNA. So there's obviously, we're not. If we looked at DNA, there'd be the whole human genome there, but this is RNA, and only 6 of the contigs were related to influenza C. And what we were looking for, in order to really be sure, and if it is influenza C, what we're looking for is we're really looking for a contig that pretty much covers the entire reference sequence and has really high aligned nucleotide identity, because then it's kind of overwhelming information that it actually is that virus. And you guys you'll see a little later on in the talk, like this paranoia is justified about these things. So and so that's what we see. We see that it's high, and then what we see is this is basically what we see. So we see we have I think 6 contigs, pretty much all of them. All of them are very, very high. They cover almost the whole. So the ANI is the aligned nucleotide identity. That's high. They cover almost the entire reference, and then they also cover the entire contig. We don't expect the whole thing because obviously the ends of the sequences are always going to be somewhat problematic. But so it's very close. So very confident that that's the case. So this looks like it really is an influenza C result. Then and so the other thing that we also did was we aligned these contigs against all of the influenza genomes in NCBI, which is like 1 million, because we're thinking maybe this isn't an influenza C, but this is actually some distant, other influenza sequence, and so maybe that could be what we're seeing. So, we aligned all the contigs against all the influenza genomes, and there's about 1 million of them. We found, 1,200 matches, and all the matches were for influenza C annotated genomes. So this is what the data looks like. And in the spirit of Ben, and actually I got this technique actually from Ryan's talk 2 years ago at CGSI. I use this tool. Does anyone know what that's doing? So I basically, send all the annotations into ChatGPT and ask what is the organism, and it says organism influenza C. So that's enough for me to agree that that is correct. Okay. So and I also want to. Okay. So I'm gonna. So but I also want to give kind of a sense of why this problem is difficult. So this is actually we have this. So from this data set, we have this positive, we have these confirmed positives, right? Clinically confirmed positives, and then we have our data, and so we can look to see, how that looks. And so this is actually for one of the viruses, parainfluenza 3, and this is the number of, so the B1, you'll see all these A1 through 12, and A through H. These are like on 96 well plates, this kind of different wells, and so a lot of our things are labeled with the wells. That makes us look like we are, very serious wet lab people. But basically, these are like the wells where there is a confirmed parainfluenza 3 sequence, and what we look at is just the number of reads that are coming from that, from that sample, with our assay, and we know that they're positives. And so this is I'm just showing you these raw numbers because the range is quite dramatic, right? The range is, look at this, it's like 100,000 difference, right, between the lowest and, this is only 10,000, but this should be 100,000, that's a typo. But anyways, it's like 100,000 increase in between E1 and H1, and these are both positives, right? So that tells you that, the range of a positive is some kind of, many degrees, many orders of magnitude. And the other issue is that even a 0.1% of contamination, like for example, if I accidentally take this well, the sample here, and I accidentally put 0.1% of that into a neighboring well, like, a little, not even a drop, but a little tiny, amount of it, it'll be higher, right, than this other true positive, right? So contamination is a real problem here. If you think about under normal circumstances, if you're looking at like total expression levels of a gene and you contaminate, 2 neighboring wells by 0.1%, then those measurements will be off by 0.1%. That's not a big deal because, your actual device is not able to measure at that level. But here, because you're really comparing against 0, and you're saying that the difference between 16 and 0 is the difference between a true positive, between a positive and negative, a tiny contamination will do that. So this is actually, and so we kind of verified this by looking at our data, because we're actually automating our assay using these automated liquid handlers at the time, and so we were verifying them by also doing manually, and we can compare them. And so if you look at these are the positions of the true position of parainfluenza, and so if you look at where our positives were for the manual workflow, you can see that there's like, it's really these neighboring wells became, these red ones are false positives, but in the automated, there's no contamination. So in fact, if you actually look at how these automated liquid handlers work, people think that they're designed to do things faster, but actually they do things much slower. And they do things much slower than any person can possibly have the patience for. And so they're actually more accurate. So, and we showed this for different viruses. Anyways, I'm just giving you in a sense that problems, because setting up for what we did this year, so we're expecting to see all sorts of these kinds of things. So this is actually what the automated liquid handler looks like when it's working at like, I think 16 times speed. It's much slower. But anyway, okay, we're gonna skip that. Okay, so here we are. And so that was actually kind of what we did last year. And this year, what we really were focusing on is this one. It worked really well, except for enterovirus, which rhinovirus is a type of enterovirus. So this this common cold, it did not work well at all. And it's not like so bad, but there's definitely a big gap between that and all the other ones which work really well. And I also had 4 new students start in the lab. And so this is actually, it's kind of sad to say, but this is kind of the only thing they worked on the whole year. Right? No, but it's true, right? It's true, yeah. But they made, yeah, they're actually sitting right here, yeah. But they made good progress, right? So there is a good story, but it's hard. And so we tried lots of different things, and I'm going to talk about some of them. So, but let me kind of take a step back and say, like, what is really the computational problem here? And so here's actually what, at least in the framework that we're doing, we have reads, and we have basically a set of reads from the sample, and then we have these large, numbers of genome sequences, and our goal is to really, for every read, at least in this framework, to classify what's its genome of origin. And so the analysis framework that we're taking is that we're really doing we're doing one read at a time. So every read is analyzed completely independently of every other read. Later, obviously, we'll count the reads, and if there's enough above a count, we will classify that that this is a positive or negative sample based on some kind of thresholds. But a key concept is that really every read is analyzed independently. I think there's an advantage that lets us kind of understand what's going on. Okay, so the other thing that we do is we also take the reads and we break them up into kmers, and we analyze each kmer independently too, right? So we take the reads, we break them into kmers, and then we build a data structure, which is basically for every kmer, we basically keep track of which genome sequence IDs the sequences are in, and so this is, and then we have this big table, and so for each read, we take every kmer, and we look up the sequences for every kmer and kind of, every kmer kind of votes, right, for which sequence is from, and that helps us, and we use that to predict the genome of origin. And this is really coming from this idea that kind of is, maybe now more than 10 years old, really from these work at Stephen Salzberg's lab on Kraken and a couple others contemporaries at the time, which were really had this kind of insight, which was saying that, you have so many reads, you have so many genomes, but if you really match something that's like 35 length and it's an exact match, and that only occurs in that one genome, that's like a good sign, right? And so, since that time, kmers have become very popular, as I think the third kmer talk at CGSI. A non-kmer person pointed that out to me today, right? But that's the basic idea. And so when does it work? So observed genomes can have mutations, and reads can have errors, and so but in order for you to be able to identify it, at least one kmer must match the read to the database sequence. But the problem is really that this table is way too large to store, and the number of kmers is really astronomical. Like in just the standard database that we use, it's 16 billion, and then also like in the standard, there's like 50,000 species. So you can imagine this table is just is huge, and so we're using a kind of a approach. This is kind of the tutorial. This is not our work. I'm really going to give you kind of how Kraken solves this, and there's many other approaches for this, but I think a lot of them share the same ideas. So one idea is that this is a kind of key idea of Kraken, which is that instead of actually storing all the sequence IDs, instead use the NCBI taxonomy and just store essentially for every kmer the node of that taxonomy that it corresponds to. And really, it's kind of like if that kmer is unique in a species, then it'll point to that species, and if it occurs in multiple species, it'll point to the least common ancestor in the tree. So things that appear in human and bacteria, for example, and in viruses, they'll all point to the root, essentially, right? So that's the idea. The nice thing about this is that, there's two nice things about this. First of all, things that are unique are stored. So anything that's unique in a species, those are all there. Things that are kind of unique to a subtree, those are also all there. But the other real advantage is that you only need to store one number for every kmer, because you have just whatever the number of the node is. So you lose information, right? Because within subtrees you don't have, but you only need one number. So that actually helps a lot. That's one idea. And the other idea, and so this is what the data looks like. You basically have, so this is actually a report from, so they actually output for you the tree, and then these numbers here, like this number here is actually how many, this is in the root, you have 3.4 million that are in the root, and then this number here is basically at that subtree. So for example, you have 3.3 million that are human, and 3.3 million are actually at that human node, and these are all the nodes basically in between, and you have, you know, that many in the subtree, but they're not at that level. You know, you have huge numbers of bacteria. In fact, you have so many bacteria that I think it's very hard to interpret Kraken results for bacteria, and so I think people like David and others, Siovash, have other tools for this. But for viruses, you know, I think Kraken actually works pretty well. And so this is what the results look like. So this is like the tree of viruses. You can see that this is somebody who has, you know, 24,000 SARS-CoV-2 viruses, and you can see that they're part of this, you know, part of this branch over here. But we also see a little bit of hits for other coronaviruses, and these are all probably false positives. They're just probably just because you have so many SARS-CoV-2, and some of them look like these other, you know, hit these other reads, but this person probably does not have a co-infection of 3 different coronaviruses, right? They probably just have one. And so, but then you also see interesting things like this tomato brown rugose fruit virus. So does anybody know what that is? Okay, so I'm hoping somebody once will reply and let me know, you know, but I have no idea, right? So, and if you look at these in Wikipedia, they're like, oh, it's like infects fruit, you know what I mean? It's not useful. Okay, so anyways, the other idea is they use minimizers, and so here's so overlap. So the idea is that overlapping kmers are redundant and shared kmers have similar sequence. So here's actually a perspective. So I actually motivate minimizers by a different problem, which is this idea of kmer counting. So if you want to count how many kmers you have, like you have a long sequence, and for every kmer you want to know how many times it occurs. You know, that's a, you know, and you want to do that over like a giant database, like, you know, all of NCBI, for example, or all of SRA hits, or, you know, this is the kind of thing that some people here work on. How would you do that? And so there's not enough, and there's so many kmers, you don't have enough memory for the actual index, and so you want to use disk to basically, you know, be able to do this computation. You know, one kind of trivial perspective is, let's say, you know, you can't store your whole index, but you can store 1/256 of your index, right? So what you could do is you could basically take the first 3 letters of the kmer and have that be the, you know, everything that follows it be an index for just that, and you'll have 256 of those indices, and you basically, you know, for every kmer you basically load up out of from disk that kmer, that index, you know, update the count and save it, right? And then go to the next kmer and keep doing that. And so if you did that, you know, for every index, you would have to, for every kmer, you would have to basically load and save, you know, the same index. It's load and save an index. So you have index swap required for every kmer, but instead you can use this concept of minimizer. So what is a minimizer? So let's say, here's an example. Here you have a 31 length kmer. Let's say you take all the 7 mers of that 31 length kmer, which are like these, and then take the one that is alphabetically, the first from the set alphabetically. Actually, I don't know if I might have, I'm looking at it. I don't see if that's there, but that's anyways, the example might be wrong, but the constant. So take all 7 mers of that, and then take the first one from the set alphabetically, and that's what you defined as your minimizer of that kmer. So this is a deterministic mapping. You'll always have, for every kmer, you'll always have the same minimizer. But, you know, and for related kmers, similar kmers, you also have the same minimizer, because especially when you shift one over, your kmer over, right, the minimizer will only change if one of two things happens. The minimizer moves out of the window or something comes in that's alphabetically ahead of it. So for many neighboring kmers, you'll have the same minimizer. So if you do that, for example, let's say you have kmers of length 100 reads. This is what the minimizer is for that kmer. Only for neighboring windows, you'll have the same minimizer, and you'll only shift when, like at this point, when actually in this case, this new sequence comes in that's ahead of it, that's alphabetically. And so if you now have a minimizer, which is your index is basically labeled by the minimizers, you'll end up only have to change your index, right, when the minimizer changes. So this is actually much faster, and I think all the kmer counting techniques use this kind of idea. So anyway, so that's the idea of minimizer. And so how does that apply? So the idea of minimizers is that instead of storing all the kmers, you basically just store the minimizers and their genome IDs, and then it works. It works. So that's I think those two things together are kind of the idea for Kraken and Kraken 2 actually specifically. Although this is they do minimizers in a different way because I'm not getting into like how you like. There's also some details on if you're going to implement this, what's the hash table data structure, and that also creates a lot of overhead, and so they handle that. So it's different, but conceptually that's the idea. Okay, so when does it work? Similar idea is that you can have reads and errors as long as the minimizers match. It's going to work. Okay, so this is the idea. So essentially, if you combine these two ideas together, you get something which is approximately Kraken 2, and then it can scale to very large databases of sequences. Okay. And so now kind of what we did. So why is this problem hard? So they're from very different sequences, so for example, if I take reads and I map them to just SARS-CoV-2, I might see 3,000 reads mapped to SARS-CoV-2. But if I take them and map them to human and SARS-CoV-2, only 2,000 reads will map. So 1,000 reads in this example map to SARS-CoV-2 only because there's no human reference sequence there. So there's a lot of similarity between viruses and human. That's one thing. And then there's this other problem, which is called reference contamination, which is that that you have these unfinished bacteria genomes, and like for example, you might have some influenza or human that's attached into your genome, and then when you map to them, you'll end up with these reads that map. I'll show some examples. Okay, so anyways, now I'll finish about 5 minutes, and it's on the organizer. I can make that executive decision, So anyways, okay. So now getting where we are. So I've set the stage. So we basically wanted to work on these enteroviruses to see if we can improve them. And again, with these our new students. And so what happened? So we only detected 27 out of 34. So possible there could be molecular causes for this, which is like low viral concentration, or maybe enteroviruses. The way that they're encapsulated, their sequences are more damaged, than they would. So there's reasons why this could be impossible, right, for us to detect, but also could be bioinformatics. And so one hypothesis is that, the circulating enteroviruses that are actually, infecting people are more distant from the sequences in the database than, let's say, the ones that for RSV, right, for example, right? So that was the hypothesis. And so the first attempt is we actually used the same data that we collected for influenza C, which is that we basically took that assembly data, which is very deep sequencing, and looked at for, in the database of enterovirus sequences, there's 10,000s of them. We identified a very close sequence in the database, verified that it's not in the RefSeq. We added to the database, so we essentially took this, the original database. We added one new enterovirus sequence, which we kind of knew was in the sample, and that essentially added new kmers, right, to our sequence. And then that actually worked. So that was good. So it's a good attempt. So essentially, if you know what you're looking for, if you can't find something you're looking for, you know what you're looking for, and then you change your method to make sure that you can find it, it'll work, right? So, but this is not really scalable because we would have to know what the virus is. So then the scaling approach is let's add all the virus sequences to the database, and then there's really some issues and errors in the database, and we'll fix this by new sequence. So here we had all the new viruses. This adds many new kmers, right? And let's see how this kind of works. Okay, so the data that we're looking at, a new set of data, which is 96 samples, 94 in the clinical. And when we apply our original method, we found essentially 3 positives. We found a SARS-CoV-2, 1 enterovirus, 1 human metapneumovirus, and then I highlight these 2 that are very weak signals, but they're below our threshold. And the idea is that we'll take the whole NT viral database and we'll add it in to our database, which is about 12 million viruses. 10 million of them are COVID. We actually don't add all of those because it would overwhelm us and it's kind of overkill. About 1 million influenza and about 1 million of everything else. So we're adding about 2 million new viruses into our sequence, which is overwhelming. In terms of it's not that much sequence because the viruses are short, but it's definitely all the species. And so what happens? Okay, so first of all, we look at these negatives and we're hoping that we find something interesting. So we look at A01, we see monkeypox. We're very excited because it's known that monkeypox can be detected using a respiratory test, but it hasn't really been shown clinically. So we're very excited that we were the first ones to detect monkeypox with this virus. Okay, so then we look. What about A02? Monkeypox, right? Very exciting. Still very excited. Okay. So, right. Okay. So literally, we see monkeypox in every sample, right? Okay. So then, this is like 2 months of Daniel's time, right? So, he looked through thousands of monkeypox sequences. He found that there's one sequence has a human rRNA just appended to it, right? And essentially was driving all of the monkeypox positives, right? But anyways, we fix this. So this is actually our, adding all the NT. We do see new things. Like for example, we only saw 575 reads. We see 141,000, right? So that clearly this is the more accurate sequence for this, right? We see, a new 3 new enterovirus sequences that we didn't see before. We see another human metapneumovirus. And then this is actually, I'm not getting into it, but we came up with a strategy to try to mask to prevent what happened before, to avoid false positives. We would actually use the clean database to try to mask out everything in what we're adding. So we're only adding sequence that's kind of new to prevent the kind of monkeypox scenario. But even still, what happened is that we had this hugely, 600,000 COVID reads for SARS-CoV-2 in our original sequence, and it disappeared in our new sequence. So what happened there? So essentially, this is our original sequence, 600 reads. In this one, we don't see it anymore. We see a lot of SARS-1 potential reads to the ancestor. So what happens is that actually, in this case, there's one COVID sequence, SARS-CoV-2 sequence, that was labeled as a the original SARS sequence. And so when it got added, it pushed all the kmers to the ancestor of that. So this is the other kind of thing that we're dealing with. And of course here, these are like examples of where I want to go through the examples of the problems because this tells you these are examples that we can really understand, and because we see, we're looking for things, but it gives you a good idea of what's actually all the problems that are going on. Anyway, okay, I'm going to end there. And so this is really work by Vivek, Joanne, Daniel, and Will in the lab, and also the lab. Okay, thank you.